Welcome back. Today we're going to talk about the two other muscle types that we haven't had a chance to discuss yet. So that's going to be cardiac muscle and smooth muscle. Going to start out with cardiac muscle. In reality, it's pretty similar to skeletal muscle in a lot of ways and different in a couple main. The reason why I say it's mostly similar is when you do look at it under the scope, you are going to see those striations again, which are very distinct and give both of them a very similar look. One of the major differences you're going to see here, and it's pointed out by some of the number of letters on here, one is the nucleus is going to be within the cells, not in the periphery, and there's generally only a single or every once in a while a couple nuclei in a cell. The other thing is these cells are branching, so you can see it a little bit in the center of the screen right here. We'll see it better on some other slides. The other thing you can see is they have these specialized structures that are in between the cells called intercalated discs, which are very rich in gap junctions and allow these cells to have a depolarization in one muscle cell influence the cell next to it. So as I was kind of saying on the last slide, cardiac muscle cells are, I wouldn't say long, but moderately long compared to a lot of cells. I mean, nowhere near as long as skeletal muscle cells are, but definitely longer than probably your average cell. We're going to see these are branched and generally have a central nucleus. Every once in a while you'll see a second one in there. Very similar to skeletal muscle cells, lots of mitochondria present, again, to allow ATP synthesis to happen very quickly, allowing these cells to be highly active. Again, the striated appearance is due to those actin and myosins kind of doing those overlapping with the thick and thin filaments to give you those, again, A and I bands that are alternating back and forth that give that striated appearance. So you can kind of see zoomed in on one of these cells here. Again, you can see branching fibers on this. That intercalated disc, you can see the gap junctions here that are connecting those to one another. And again, really means that a depolarization in one cell is going to travel and influence the one next to it. So all the cells that are connected to one another are actually going to act as a single unit, which is not the case in skeletal muscle, where each cell acts only as it's by itself in skeletal muscle. But outside of that, a few other things that are similar: transverse tubules, the myofibrils are going to look somewhat similar. Again, that overlapping pattern within the sarcomeres that gives that distinct striated look. So, not quite as common in the cardiac muscle is that sarcoplasmic reticulum. If you recall, it was used to store intracellular calcium in those cells. A lot of the calcium that's going to be, it still does do that in the cardiac muscle, but the vast majority of calcium that's going to be involved with the actin and myosin interaction is actually coming extracellularly from the outside of the cell. And again, T-tubules, where they're coming in there, is a little bit different than what we see in the skeletal muscle. Again, not something that I'm that worried about. To me, the biggest thing going on with this, and that one of the things that makes them distinct, and depending on which digital slide you look at, sometimes it's stained for these structures, sometimes it's not, are these intercalated discs. These are specialized connections between each of these cells. They have a couple of the different types of cell adhering structures there, so those fascia adherens and the macula adherens, those desmosomes and other stuff like that were actually holding the cells together well. Again, this is an organ that is going to fill up with blood and then it's going to contract, so you want those cells to be well connected to one another, so when that heart undergoes that forceful contraction that you do not separate from in between those cells. The other thing we're going to see at these intercalated discs are going to be the presence of those gap junctions, and again, without getting into too much detail for the histology course here, this is going to allow that depolarization that was going to travel all over like the skeletal muscle cell, but stay within that cell only when it depolarizes, is in skeletal and cardiac muscle is going to actually allow that depolarization that starts in one cell to travel to all the cells that are connected to it. And again, just kind of showing you a cartoon version of that intercalated disc structure. And again, as you can see here, branching fibers, centralized nucleus, intercalated disc. Those are the things that are going to give you that distinct different from that skeletal muscle. Again, it does have the striations, which is something we are not going to see in the last one that we're going to talk about, which is going to be the smooth muscle. One of the things you'll see if you're looking overall at the tissue, and I don't try to make you identify this when we're talking about just the muscle, is there is going to be a very good blood supply to cardiac muscle. Cardiac muscle is one of those things that, because it is the pumping of the heart, we're going to have extremely good blood supply to the heart, so it always stays in a aerobic type of respiration, that the heart is never starved for oxygen, and if it is, that is a lot of times what's going to be causing a heart attack or something bad going on with a cardiac event. The other thing we're going to see is the structures. If you think about the heart, they kind of spiral down to the tip and then go back up the other side. And what this does is when those fibers contract, when we start thinking about a heart, it's not just a squeeze in or a squeeze up. It's kind of an in and up, which is going to be the most efficient type of a contraction to get the most blood out of the heart for that particular amount of force. All right, so before I do the slides, the other thing I wanted to talk about real quick was smooth muscle, and then we will again go to the digital slides, and I'll show you examples of both of those digital slides with some of those structures present in them. Smooth muscle is probably, in terms of where it's at, is a little bit more widespread than the other two. Again, cardiac muscle only in the heart. Skeletal muscle is those voluntary muscles that you have control over, things like the bicep, tricep, a amount of other muscles we can name. Where smooth muscle is going to be a lot of times called organ muscle, visceral muscle. It is going to be lining a lot of your digestive tract or other muscular organs like the gallbladder, but is also present in the iris in the eye, present in the erector pili muscles in the skin that makes your hair stand up, gives you goosebumps. So we're going to see this found in a lot of different areas. One of the things you can see a lot of times, we will see it in two different orientations, especially with the digestive system. So a lot of times you'll see this on a slide. You'll see it longitudinally in one way here. Again, more what I want you to be able to identify the difference on. But a lot of times, anytime you're looking at smooth muscle, a lot of times it's going to be a digestive tract organ that we're going to see it in. So the small intestine, large intestine, esophagus, something like that, we'll find this smooth muscle in. A lot of those tubes, you're going to have two different structures. So if you think about like this pencil being the inside of the tube, the inner layer of muscle on this one, of that muscularis, is always going to be a circular one that goes around it like this. We then have another layer that is kind of sitting on the outside, this longitudinal muscle that allows us to do that peristaltic action, which is shortening of the tube in front of wherever the bolus is traveling, and then a squeezing in behind whatever the bolus is traveling to keep it moving down that tube. So a lot of times, any slide you look at, you will see both these orientations there. Again, as I was already saying, lining a lot of these muscular organs, things like the intestines, the stomach, uterus is another good example on this one. Again, found in the blood vessels, found in the eye, as I mentioned. Again, found in a lot of different organs. We're going to see there is some other structures that are around there that help connect these things down to where they're, to give them the strength and the connectiveness to the cells to one another. So these organs, much like other ones, are not going to pull themselves apart at all when they contract. One of the things that's interesting about these ones is these are the only form that's not striated. I always kind of say the easiest thing to actually honestly confuse this slide for is dense regular connective tissue. That dense regular connective tissue had all those fibers traveling in the same direction with nuclei of different fibroblasts in between. These ones, you're going to look like you have a bunch of fibers going in the same direction. In reality, they are these spindle-shaped fibers that you can see on the screen with a centralized nucleus. But again, if you mesh them all together, like I'm doing with my fingers here, they make a sheet of muscle. And again, these ones are going to contract a little bit differently. They don't just get shorter, they get shorter but also narrower because all the fibers aren't traveling in the same direction, which is why they don't have those striations. Again, generally a centralized nucleus, and also very tightly packed one under these cells you're going to find. Again, decent amount of mitochondria and other structures that are going to be in these ones here. They're generally going to be surrounded by some reticular fibers around each side here. And again, still made up of actin and myosin in terms of their myofilaments. But they're going in a number of different directions within the cell. So you don't get that regular pattern where all those things were lining up to give you that striation. And we'll see how they're kind of connected in there. You're going to have a lot more filaments that are helping to hold these things together and in place. So you can see with smooth muscle, you can see kind of a zoom in of these filaments here. But you can see over on the actual spindle-shaped cells here, we have what are called dense bodies. These are kind of junctions between these different filaments. And what we're going to be doing is having these actinomyosin filaments in between these dense bodies, which are going to be traveling in a number of different directions. And when you think about the cell overall, it's going to squeeze it down in terms of length, but also in terms of width, so that whole sheet is going to kind of change in size and get smaller, both in length and width. The other thing you notice is kind of the overlap here. This figure is actually not correct. There's actually myosin heads along the whole length of the thick filaments here, which means that these things can contract more forcefully. They don't have a length relationship to how strong they can be. The other thing that's interesting about smooth muscle is it can contract and stay contracted without using a lot of ATP, which is something your skeletal muscle can't do. If you ever take A&P, we'll talk about it in there. It's called latch bridge mechanism, but that is why you're able to have sphincters, like things in your stomach and other parts of your body that hold that sphincter close. So think like the urinary sphincter, that hold that closed without running out of energy, without fatiguing, which in a lot of these sphincters is not something we'd want to have happen. Again, those dense bodies, they're really acting as the ends of the particular sarcomeres. And so instead of Z lines, we don't have it here. We have these dense bodies where those thick and thin filaments are going to be pulling those dense bodies closer to one another. And again, found within the cytoplasm of these cells, as well as attached to the membrane, which is going to allow these cells to get kind of pulled in a number of different directions. And again, you can kind of see it in this picture right here, showing a relaxed smooth muscle cell, and how this picture right here shows that it did get narrower as well as shorter, which is showing us it changing in both those directions. So just to remind everybody, we talked about 3 different muscle types again. We had skeletal muscle, cardiac muscle, and smooth muscle. We're going to see there is some limited ability of these to undergo some regeneration. When we talked about skeletal muscle in the first show, we really kind of saw that it's honestly extremely limited. You can strengthen the muscle fibers that are left, but if you lose some muscle fibers, you're not going to lay down new muscle fibers or new muscle cells, which, again, interchangeable words there. Cardiac muscle, similarly, does not have really any regenerative abilities. And early on in childhood, there is a little bit more plasticity in those cells. You might have a little bit of regeneration of some damage to heart tissue in somebody that's incredibly young, but as an adult, if you have damage to the heart muscle, it generally does not get fixed. It becomes scar tissue, which again is connective tissue. The last type, smooth muscle, it does actually have the ability to undergo regeneration. These ones can divide and then can replace damaged tissue. So it's really the only muscle type that we see that happening in. Look at some of the digital slides. You can kind of see this is a large section of skeletal muscle. I'm going to zoom in on a particular area, which was actually right over here. This one to me is kind of the area, and this is what you want to try to find on one of these, is a little bit more of a longitudinal slice on these ones, because again, that's how I want you to be able to identify these. If you're looking at this one, you can notice a few different structures as we're looking at it. First off, you can see the nuclei are very prominent. So, and again, you can see a branch fiber right here, centralized nucleus in these ones. The striations are not super clear. I'm having to look a little closer at the screen, but you can see some striations where I have the cursor right now. The other thing you can see is in between some of these cells, you're seeing these intercalated discs. That's that dark line structure there. There's a couple of good examples of intercalated discs over here and over here. So to me, those are the main things to be looking at, and honestly, the things you're going to need to be labeling as you look at the cardiac muscle. Again, knowing where it's at, knowing what it does well, are all things that we talked about before with that type of stuff. So the other slide that we're going to need to take a look at here real quick is some type of smooth muscle. So let's take a look at the smooth muscle on this one. So on this is one, you can kind of see, if you're looking at the side of the screen over here, this is an intestine with a lumen right there, What we're going to be looking at is the muscle that's on the outside part of this. Somewhere down the line, we'll be talking about all these different layers in a week or so here, a couple weeks when we get to the digestive system. But to start with, we just want to go to this outer part here. And as we zoom in on this a little bit, well, first off, let me point out a couple things because I kind of put you right on the cut line there. You can see this is that inner circular layer that we're going to see it longitudinally. Down beneath that is, the inner circular layer, and then the outer longitudinal layer is what we're seeing cutting through it in a transverse type of section. So in terms of identifying it, I'm not going to show you this bottom part here, but this top part is definitely going to be something that would be fair game. So let's find an area that has a nice longitudinal section, which all this looks pretty good here. So what we'll do now is we'll zoom in on this a little bit more. And one of the things you notice on this one, you don't see the striations. You're not seeing a lot of other structures. The main thing you see on this one is you can kind of see that spindle-shaped cell right here with a centralized nucleus. The big thing to look for if we're looking at stuff like this versus, let's say, the dense regular connective tissue. The difference you're looking for is you can see on these ones, the nuclei tend to be in the center of these cells, where when we were looking at, let's say, like a tendon, you were seeing it on the periphery of those cells, of the fibers, not in the center of a cell. And to me, that's the main thing to look for and go after on this one. So this one, being able to identify it as smooth muscle, the nucleus, that spindle-shaped cell, the lack of striations, those are all the things that are going to help you identify this as smooth muscle. I'll see you again next time when we start talking about some of the other tissue types and moving on from this chapter to nervous tissue.